Deep beneath the ocean, there are creatures that look almost familiar, but exist in conditions nothing familiar could survive. Right now, on the cold, dark seafloor, giant isopods move slowly through crushing pressure, long periods without food, and an environment where light never reaches. Tonight, we'll explore what deep-sea isopods are, how they survive these extreme conditions, how they feed, grow, and adapt, and what they reveal about life in the deepest parts of the ocean. You don't need to keep track of anything. This will unfold on its own. We'll begin with what an isopod actually is, and how something so ancient still exists today. Isopods are crustaceans, members of the same family that includes crabs, shrimp, and lobsters. They carry that lineage in every joint of their body, in the way their legs bend and their shell curves to hold them together. The crustacean body plan is one of nature's most enduring designs, structurally sound and biologically versatile enough that the ocean has maintained it for hundreds of millions of years. The isopod wears that inheritance quietly, moving through the deep as though it has always known exactly where it belongs. In the cold and the dark, the isopod is not a stranger. It is a native. The order Isopoda contains more than 10,000 known species, making it one of the most diverse crustacean groups on Earth. Some are no larger than a grain of rice. Others grow to the size of a dinner plate. Some live in the canopy of tropical forests. Others drift in freshwater streams or burrow beneath sandy tidal flats. Each species represents a variation on a shared body plan, reshaped by time and environment into something slightly different. The deep-sea isopods are one chapter in this broad story, among the most extraordinary of the group, adapted to a world of cold, pressure, and permanent darkness. The name isopod comes from the Greek words isos, meaning equal, and paus, meaning foot. Together, the name means equal feet. All 14 of their legs are roughly the same size and shape, unlike the varied limbs of a crab or the large claws of a lobster. Each limb performs the same function, and they move together in a coordinated rolling rhythm that propels the animal slowly and steadily across whatever surface lies beneath it. The uniformity of the isopod's limbs is a structural feature, not an aesthetic one, but it does give the animal a distinctive, deliberate quality of movement that is unlike almost anything else in the deep sea. Isopods are found in nearly every habitat on Earth, from rain-soaked forests and underground caves to freshwater rivers, rocky coastlines, and the deepest ocean trenches ever explored. The common pillbug found in gardens is an isopod. So is the creature moving slowly 2,000 meters beneath the surface of the Atlantic. The same body plan, distributed across nearly every environment the planet offers. This range is a measure of the group's adaptability. Not a single environment has proved definitively inhospitable to isopods as a whole. Wherever a quiet corner exists, some member of this order has found it and made use of it. The deep-sea isopods that have received the most scientific attention belong primarily to the family Chirolanidae and the genus Bathynomus, among others. Within the genus Bathynomus live the largest and most studied of all deep-sea isopod species, animals that have become, in a sense, the most visible representatives of the deep ocean floor to the broader public. To know the genus Bathynomus is to hold a reference point for the kind of life that exists far below the reach of sunlight, at depths where pressure alone would be lethal to most other animals. The giant isopod, Bathynomus giganteus, is the most well-known deep-sea species. It was first formally described by French zoologist Alphonse Milne Edwards in 1879, following the collection of specimens during deep-sea dredging expeditions in the Gulf of Mexico. Milne Edwards encountered something unlike any known species at the time, a large, heavily armoured crustacean pulled up from depths that were, in the scientific understanding of the era, thought to be largely uninhabited. He gave it a name that fit its form, Giganteus. The description he published opened the first formal scientific record of giant isopod biology and initiated over a century of continuing research. Isopods have been present on Earth for at least 300 million years. Fossil records trace them back to the Carboniferous period, a time when large fern forests covered much of the land and the earliest reptiles were only beginning to appear. 300 million years spans the rise and fall of numerous species, the movement of continents, and the emergence and extinction of entire ecosystems. Through all of it, isopods persisted, not through dominance or rapid adaptation, but through the kind of structural reliability that means a body plan remains functional across an enormous range of conditions. The isopod is among the oldest continuously existing body plans in the animal kingdom. Each of the isopod's 14 legs ends in a small curved structure called a dactylus, a claw-like tip that allows the animal to grip uneven surfaces on the ocean floor. Rocky outcroppings, crusted sediment, the bones of a sunken carcass. The dactylus finds purchase across all of these substrates. This gripping capacity is particularly valuable in an environment where slow, deep currents can exert lateral force on a stationary animal. The dactylus is a small feature by any measure, but it represents the final point of contact between the isopod and the surface it depends on, a detail that matters considerably when the alternative is being displaced from the seafloor entirely. Giant isopods have two large compound eyes, each composed of hundreds of individual lenses packed together in a structure called an ommatidium array. This arrangement does not produce a single focused image the way a human eye does. Instead, it assembles a wide, mosaic-like field of visual information that is particularly sensitive to movement and changes in light levels. In the deep sea, where usable sunlight is entirely absent, these eyes are calibrated not for detail but for detection, the ability to register the faint bioluminescent light produced by other organisms moving through the dark water. The compound eye, in this environment, functions as a motion and light detector more than a conventional visual organ. The compound eyes of giant isopods are sessile, fixed directly into the surface of the head rather than mounted on movable stalks, as they are in crabs and many shrimp species. The isopod cannot redirect its gaze or pivot to track movement in its peripheral field. Its visual range is fixed and determined entirely by where the animal itself is facing. This limitation places greater functional weight on the isopod's other sensory systems, particularly its antennae and chemoreceptors, which compensate for the restricted field of vision. The fixed eye does not pursue information. It receives what the environment delivers within its static field of view. Isopods have two pairs of antennae, one shorter pair and one longer, which serve as the animal's primary instruments for reading the surrounding environment. In an environment without usable light, the antennae carry an outsized sensory burden. They sample the chemical composition of water passing across them continuously, detecting dissolved organic compounds at concentrations that human instruments would struggle to measure. They also detect mechanical vibration, the pressure waves generated by movement nearby, and respond to direct physical contact by registering the texture and form of whatever surface they encounter. The antennae are how the isopod navigates, locates food, and senses the approach of other animals, all without the use of vision. When threatened, a giant isopod can roll its entire body into a tight, nearly spherical shape, drawing its legs inward and enclosing its soft underside within the hardened shell. This defensive behavior is called conglobation, and it is among the oldest documented anti-predator strategies in the arthropod lineage. The common pill bug uses the same reflex, inherited across hundreds of millions of years from a shared ancestor. In the deep sea, where the nature of a threat may take unfamiliar forms, conglobation provides a reliable last resort, converting the animal into a compact, armored sphere that presents no exposed soft tissue to whatever has triggered the response. The exoskeleton of an isopod does not grow with the animal. It must be shed and replaced periodically in a process called ecdysis, or molting. What distinguishes the isopod's molt from that of most other crustaceans is that it occurs in two separate phases. First, the posterior half of the body sheds its old shell and expands into a new, larger one. Sometime later, the anterior half follows. For a brief interval between phases, the animal's front and back halves are at different stages of the molt, old shell on the front, new shell on the back. During this transitional period, the isopod is at its most physically vulnerable, with partially exposed soft tissue awaiting the completion of the second phase. The coloration of giant isopods ranges from pale grey to soft lilac. In the total darkness of the deep sea, colour has no functional role in camouflage or species recognition, as no predator or potential mate can perceive it visually. The pale tones of Bathynomus giganteus are largely the result of reduced pigmentation, a consequence of the metabolic economics of deep sea life rather than any active adaptation to the environment. Color for the deep sea isopod is an incidental feature of its biology. It exists in a place where it serves no purpose and is witnessed by no one. Despite their size, giant isopods are relatively light in proportion to their volume. Much of the internal architecture of their shell is hollow, reinforced with structural ridges rather than filled with solid material. The design is oriented toward pressure resistance rather than mass, distributing the mechanical load of deep water across a network of curved surfaces and overlapping plates rather than resisting it through sheer material density. The result is a body that can withstand the pressure of 2 kilometers of water without being made correspondingly heavy. The isopod's shell is a study in structural efficiency, minimal material, maximum resistance, refined across hundreds of millions of years of deep sea pressure. Deep sea isopods typically inhabit depths between 170 meters and 2,500 meters below the ocean surface. Within this range, conditions change progressively with depth. Usable sunlight disappears within the first few hundred meters. Temperature drops steadily toward freezing. Pressure increases by roughly 1 atmosphere for every 10 meters of descent. By the time the seafloor appears at these depths, the environment is cold, dark, and under enormous pressure, conditions that exclude the vast majority of animals but the deep sea isopods have occupied continuously for hundreds of millions of years. Bathynomus giganteus is most commonly found in the Atlantic Ocean, particularly in the Gulf of Mexico and along the eastern continental shelf of the United States. These are waters relatively close to human coastlines, crossed daily by cargo vessels, fishing boats, and research ships operating at the surface. The seafloor beneath those ships, at depths of several hundred to several thousand meters, is the isopod's habitat. The two worlds are separated not by distance but by depth, by a column of water that, within a few hundred meters, becomes an environment as foreign to surface conditions as any place on the planet. The deep sea environment inhabited by giant isopods spans 2 major oceanographic zones, the bathypelagic zone, which begins at approximately 1,000 meters, and the abyssal zone below. Both are regions of permanent darkness, near freezing temperatures, and pressures that would be immediately lethal to unprotected humans. Scientists classify these as extreme environments. For the isopod, the classification is a matter of perspective. These are the only conditions the species has ever known. The temperature, pressure, and darkness are not exceptions to be survived, but the baseline around which the animal's entire biology has been organized over millions of years of adaptation. At a depth of 2,000 meters, the hydrostatic pressure pressing against every surface is approximately 200 times greater than atmospheric pressure at sea level. At that pressure, a human body without mechanical protection would be compressed beyond any recognizable form almost instantly. The isopod rests in that pressure without measurable physical effect because its body was shaped by that pressure over evolutionary time. Its shell distributes force across curved and overlapping surfaces rather than bearing it at any single point. The isopod does not resist the mechanical demands of its habitat. Its anatomy reflects millions of years of accommodation to them. The isopod's body resists deep sea pressure not through rigid mass but through structural geometry. Force applied to its shell is distributed across multiple curved and overlapping segments simultaneously, so that no single point absorbs a disproportionate load. This principle, load distribution across a curved segmented surface, is also used in the engineering of bridges, pressure vessels, and aircraft hulls. In the isopod's case, the solution was arrived at through natural selection rather than calculation over a timescale that engineering cannot replicate. Every arch in the isopod's shell, every overlap of one plate upon the next, reflects a structural response to the demands of extreme depth. The sea floor where isopods live belongs to what oceanographers call the benthic zone, the bottom layer of the ocean characterized by cold sediment, hard substrate, and near total stillness. The benthic zone is not a dynamic environment in the way that shallower waters are. Currents are slow. Food arrives infrequently and from above, drifting down from the upper ocean over long periods. The sediment accumulates gradually, layer by layer, incorporating the organic material of millions of years of marine life. Isopods move across this floor as functional participants in its ecology, consuming what accumulates there and processing it back into forms the environment can use. Water temperatures in the deep sea habitat of giant isopods typically range between 1 and 4 degrees Celsius year-round, just above freezing without seasonal variation. This cold is not a hardship the isopod manages. It is the thermal environment in which all of the isopod's biological processes have been calibrated over evolutionary time: its metabolism, growth rate, healing speed, and cellular function. The near-freezing temperature is what allows the isopod to run all of those processes at a low energy cost. Cold water in this context is not a constraint, it is the setting at which this particular biology operates most efficiently. No sunlight reaches the depths where giant isopods live. Usable solar light disappears within a few hundred meters of the ocean's surface. Below that point, the only light present is bioluminescence, the cold light produced by living organisms through chemical reactions in their own bodies. Bacteria, jellyfish, fish, squid, and many other deep-sea animals produce bioluminescent light, creating brief and localized illumination in an otherwise lightless environment. This is the light that the isopod's compound eyes are calibrated to detect. It is not the continuous ambient light of a sunlit world, it is intermittent, dim, and produced entirely by the organisms that share the isopod's habitat. The deep ocean floor where isopods live is one of the least explored environments on Earth. Scientists estimate that more than 80% of the world's oceans remain unmapped, unsampled, and largely unstudied. Humans have sent spacecraft to the Moon and Mars and have mapped the surfaces of Venus and Mercury through instruments and probes. The seafloor beneath the world's oceans remains, in large portions, less documented than any of those distant surfaces. The isopod lives in territory that science has only begun to examine in any systematic way, and each survey of the deep ocean floor continues to produce species, behaviors, and ecological relationships that were previously unknown. While Bathynomus giganteus and other large isopod species are generally restricted to bathyal depths of up to around 2,500 meters, other members of the broader order Isopoda, particularly species within the suborder Acelota, have been documented in the Hadal zone, which encompasses the deepest oceanic trenches at depths exceeding 8,000 meters. These Hadal isopods are physically quite different from the giant species, smaller, more delicate in form, and adapted to the even more extreme pressure and cold of trench environments. They are not the same animals as Bathynomus, but they demonstrate that the broader isopod lineage has colonized the full vertical range of the ocean floor, from shallow coastal margins to the deepest points on Earth. Giant isopods are found across a range of seafloor substrates, soft muddy or sandy sediment, where organic material accumulates and can be examined, and harder rocky surfaces, which may concentrate food and provide stable resting points. They do not confine themselves to a single microhabitat type, but move between substrates as the seafloor changes beneath them. This flexibility reflects a generalist approach to habitat use that is consistent with the isopod's broader ecological role as an opportunistic scavenger. The animal does not require specific substrate conditions. It navigates whatever the seafloor presents and extracts what it can from the available environment. Giant isopods are primarily scavengers, feeding on the carcasses of dead whales, fish, and squid that sink from the upper ocean to the seafloor. Oceanographers use the term marine snow to describe the continuous slow drift of fine organic particles from the surface downward, and whalefall for the more substantial arrival of a large carcass. A whalefall delivers an enormous concentration of organic material to the seafloor in a single event, a sudden and significant food source in an environment where nutrition is otherwise scarce and unpredictable. Isopods are among the first animals to detect and arrive at these sites, drawn by chemical signals that travel through the water column ahead of the carcass itself. Beyond scavenging, giant isopods are also active predators when conditions allow. They will consume slow-moving prey including sea cucumbers, sponges, nematodes, and small or weakened fish. In a food-scarce environment, the ability to shift between scavenging and predation provides a meaningful advantage. The animal is not dependent on a single food source or on the timing of carcass arrivals. This behavioral flexibility is consistent with the isopod's broader ecological profile as a generalist, an animal that has evolved to make use of whatever nutritional resources the deep sea provides, in whatever form and at whatever intervals they appear. Isopods are equipped with 4 sets of specialized mouthparts that work in combination during feeding. The mandibles, which perform the primary work of breaking down hard or dense material, the maxillae, which assist in manipulating and filtering food particles, and the maxillipeds, which help position and hold material during the feeding process. Together, these structures allow the isopod to process a wide variety of food types, from soft decaying tissue to harder organic material, with considerable efficiency. The mouthparts are compact and protected within the cephalon when not in use, deployed only when the animal has encountered a food source and begun to feed. When food becomes available, giant isopods are capable of consuming very large quantities relative to their body size. They feed intensively, filling internal storage capacity to its functional limit. This behavior is an adaptation to the unpredictability of deep-sea food availability. The animal banks energy during periods of abundance to sustain itself through the extended periods of scarcity that typically follow. The feeding behavior of the giant isopod is not incidental. It reflects a well-established ecological strategy in which the timing and volume of food intake are managed in direct response to the feast or famine conditions of the benthic environment. In one of the most extensively documented cases of fasting in the history of zoological observation, a captive giant isopod held at a Japanese aquarium survived without eating for over 5 years, 5 years and 4 months to be precise, before dying in 2014. The animal showed no measurable decline in outward health for the duration of the fast and refused all food offered by aquarium staff throughout that period. The case attracted international scientific attention and remains, to date, one of the longest documented fasting periods recorded for any animal of comparable size in a controlled environment. No fully satisfactory explanation for the duration of the fast has been established. The isopod's capacity for extended fasting is understood to be an evolutionary adaptation to the food supply conditions of the deep sea. In an environment where a significant food source may arrive at intervals measured in months or years, an animal whose survival depends on regular feeding is at a fundamental disadvantage. Over millions of years, natural selection in deep-sea isopod populations favored individuals whose metabolic processes could slow to the minimum rate required to sustain life and maintain that rate for extended periods without deterioration. The fasting ability of the giant isopod is not a curiosity, it is a core feature of the animal's ecological fitness in its native environment. The digestive system of the giant isopod is highly efficient, capable of extracting nutritional value from organic material that has already begun to decay or has been partially processed by bacterial activity over time. Other animals may pass over material in this condition as nutritionally marginal. The isopod's digestive system is adapted to extract whatever is available from it, drawing energy from molecules at concentrations and in chemical forms that a less specialized system would not process effectively. This efficiency is consistent with the animal's broader biology, which is oriented throughout toward extracting the maximum return from minimal inputs. Isopods occupy an important functional role in the deep-sea food web as decomposers and nutrient recyclers. By consuming decaying organic matter and processing it through their digestive systems, they return nutrients to the seafloor sediment in forms accessible to bacteria, worms, and other invertebrates. Without this processing activity, organic carbon and nitrogen would accumulate in the benthic zone in locked, unavailable chemical forms. The isopod's feeding activity is, in this sense, a service to the broader ecology of the deep ocean floor, not a conspicuous or dramatic one, but a structurally necessary one. The deep sea's chemistry depends in part on the isopod doing what it has always done. Deep sea footage captured by remotely operated vehicles has documented giant isopods gathering in substantial numbers at whale carcasses, arriving within hours of the carcass reaching the seafloor and feeding in dense groups that strip soft tissue from bone with systematic efficiency. These observations have been among the most widely viewed recordings from the deep ocean, introducing a broad public audience to the ecological activity of the benthic zone. From a scientific standpoint, the footage provided direct observational evidence of isopod feeding behavior, arrival timing, group dynamics at food sources, and the speed of detection, data that had previously been difficult to obtain through specimen collection alone. Despite their size and the precision of their mouthparts, giant isopods pose no physical threat to humans. Their claws and mandibles are adapted for soft organic material, decaying tissue, sea cucumbers, sponge, and are not capable of inflicting meaningful injury. Researchers who have handled live specimens describe them as passive and non-aggressive. The animals show no defensive behavior directed at humans during handling, beyond the standard conglobation response that serves as a general-purpose defensive posture. This passivity is consistent with the ecological context in which the isopod evolved, a deep-sea environment where the threats it faces are very different from those posed by a human hand. The organic material contained in a single large whale carcass, a blue whale for instance, which may weigh up to 150,000 kilograms, is sufficient to sustain a deep-sea benthic community for decades. Isopods are among the earliest and most active participants in this decomposition process, but the ecological succession that follows a whale fall eventually involves hagfish, sleeper sharks, polychaete worms, and specialized bacterial communities that consume what the larger animals leave behind. A whale fall functions as a temporary island of concentrated biological resources in an otherwise nutrient-poor landscape, one that the deep-sea community, including the isopod, has evolved to exploit with considerable efficiency. Giant isopods are slow-moving animals. Their pace reflects the metabolic economics of life in the deep sea, where food is scarce and every calorie expended on movement must be justified by the probability of a return. In an environment that rewards energy conservation above almost all else, the isopod's slow gait is a functional adaptation rather than a physical limitation. Observations from deep-sea camera deployments consistently show individuals moving at a measured, deliberate pace, covering ground steadily but without urgency, pausing frequently and resuming movement at intervals that suggest the expenditure of effort is being carefully managed. Isopods are primarily solitary animals. They move through their environment alone, without coordinating with others of their species or forming stable social groups. The deep sea does not favor sociality in most benthic organisms. Food sources are dispersed and unpredictable, and there is no clear functional advantage to group travel in an environment where individuals are spaced widely across a large and featureless substrate. Isopods do aggregate at food sources when those sources are large enough to support multiple individuals simultaneously, but these gatherings are opportunistic rather than organized. When the food is consumed, individuals disperse and resume solitary movement. Giant isopods are benthic organisms, animals whose lives are organized around the seafloor rather than the open water column above it. They walk rather than swim. They inhabit the bottom rather than drifting through mid-water. This attachment to the substrate is reflected throughout the isopod's anatomy, in the gripping structure of the dactylus at the end of each leg, in the flattened ventral profile suited to movement across a surface, and in the sensory systems oriented toward detecting chemical signals and vibrations carried through the seafloor sediment. Juvenile isopods may spend time in the water column during early life, but adult individuals are consistently found on or immediately adjacent to the bottom. Isopods are capable of short-distance swimming using their pleopods, the paddle-like abdominal appendages that normally function as gills. When swimming, these structures beat in a coordinated rhythm that lifts the animal off the sea floor and propels it a limited distance through open water. Swimming appears to be used for repositioning, moving to a new area of substrate, clearing an obstacle, or responding to a chemical signal arriving from a direction not accessible by walking. It is not a sustained mode of locomotion. The effort is brief, the distance modest, and the animal returns to the seafloor as soon as the repositioning is complete. Research observations suggest that giant isopods are most active under low-current, undisturbed conditions, periods when water movement in their immediate environment is minimal and the seafloor is relatively still. This preference is consistent with the isopod's sensory profile. Its primary means of detecting food and navigating its environment are chemosensory and vibrational, both of which require stable water conditions to function effectively. In turbulent or disturbed water, chemical gradients are disrupted and vibrational signals become difficult to interpret. The isopod's preference for calm conditions reflects the operational requirements of the sensory systems it depends on most heavily. When resting, giant isopods often remain motionless for extended periods, sometimes partially buried in soft sediment with only the dorsal surface of the shell exposed. This behavior serves multiple functions at once. It reduces energy expenditure during periods when movement is not warranted, it lowers the animal's physical profile against the substrate, and it places the ventral sensory surfaces in direct contact with the sediment, a source of chemical information about recent organic activity nearby. A resting isopod is not inactive in a functional sense. It is stationary but attentive, processing information from the environment while expending as little energy as possible doing so. The gait of the giant isopod across the seafloor, slow, measured, with frequent pauses between steps, is consistent across all documented observations of the species in its natural habitat. Each leg is placed with care, and the animal moves without apparent urgency, regardless of context. This pace is not simply a product of the cold temperature slowing the animal's physiology, though cold does lower metabolic rate. It reflects a deeper behavioral orientation toward energy conservation that runs throughout the isopod's activity profile. The isopod moves at the rate that the deep sea has selected for over millions of years, slowly enough to survive on very little, consistently enough to cover ground. Isopods are sensitive to mechanical vibration, detecting the pressure waves generated by movement in the surrounding water through mechanoreceptors distributed across the body surface. These receptors can register the approach of large animals from a distance, including the slow, low-frequency vibrations produced by a large carcass settling toward the seafloor or by a significant predator moving across the substrate nearby. In a lightless environment where vision is limited and acoustic signals are absent in any conventional sense, mechanoreception provides a critical early warning function. The isopod reads the physical state of the water around it continuously, using vibration as a primary channel for information about what is nearby. Studies using baited camera traps deployed on the seafloor have shown that giant isopods can locate and arrive at food sources within hours of placement. The distances implied by these arrival times indicate that the animals detected the food source from a significant distance, navigating toward it through a chemical gradient in the water. This detection capability speaks to the sensitivity of the isopod's chemosensory systems, the antennae and distributed chemical receptors that allow it to identify and follow dissolved organic compounds across the open seafloor. In a habitat where a meaningful food source may appear anywhere within a large area, this long-range chemical detection is among the most valuable functional adaptations the isopod possesses. Giant isopods show no evidence of complex social behavior, communication systems, or stable hierarchies of any kind. Interactions between individuals are limited and transient. Brief convergences at food sources, occasional encounters during movement, and the infrequent contact associated with reproduction. There is no documented territorial behavior, no cooperative feeding strategy, and no recognizable form of inter-individual communication beyond the chemical signals associated with mating. The isopod's behavioral repertoire is narrow relative to many other animals of comparable size. Its existence is organized around individual survival, finding food, conserving energy, reproducing when conditions permit, and continuing to move across the sea floor. Observations of giant isopods in aquarium environments have noted a pattern of deliberate investigation of unfamiliar objects introduced into their enclosures. Individuals approach new objects with antennae extended and actively contacting the surface, spending time in close proximity before moving away. Whether this constitutes curiosity in any cognitive sense comparable to vertebrate behavior is not established. What the observations do indicate is that the isopod's sensory systems are actively engaged by novelty in the environment, and that the animal devotes time and sensory attention to objects or conditions that differ from the baseline of its surroundings. This behavior has been observed consistently enough across individuals to be considered characteristic of the species. Female giant isopods carry developing eggs in a specialized structure called a marsupium, a ventilated brood pouch located on the underside of the thorax between the walking legs. The marsupium provides physical protection for the eggs throughout development, shielding them from mechanical damage and regulating the flow of oxygenated water across them. It is not simply a container, but a functional incubation structure. maintaining the conditions necessary for egg development within the thermal and chemical environment of the deep sea. The female carries the marsupium throughout the brooding period, with the developing eggs held close to her own body and protected by the same exoskeleton that protects her. The eggs of giant isopods are among the largest produced by any marine invertebrate, with individual eggs measuring up to 13 millimeters in diameter, approximately the size of a blueberry. This large egg size reflects the isopod's reproductive strategy, which prioritizes a small number of well-provisioned offspring over the production of large numbers of small eggs released to open water. Each egg contains substantial stored energy, sufficient to support the full development of a juvenile isopod within the marsupium without external food input. The large size of the egg is directly related to the completeness of the juvenile that eventually emerges from it, an animal that is fully formed and immediately capable of independent life. Female giant isopods carry relatively small clutches, typically between 9 and 30 eggs at a time. This number stands in sharp contrast to the reproductive output of most marine invertebrates, which may release hundreds of thousands or millions of small eggs into the water column during a single spawning event. The isopod's low clutch size is the direct counterpart of its large egg size and extended brooding period. Each egg represents a proportionally larger investment of biological resources than any single egg in a mass spawning strategy, and each is given the full benefit of the marsupium's protective and oxygenating function for the duration of its development. Giant isopods undergo direct development. They do not pass through larval stages as most marine animals do. There is no free-swimming larval form, no series of morphological transformations, and no extended planktonic phase. Instead, the young emerge from the marsupium as manchae, juveniles that are structurally complete and physically recognizable as isopods from the moment of emergence. This mode of development is relatively uncommon among marine invertebrates and is associated with reproductive strategies that emphasize the survival of individual offspring over reproductive volume. The mancha that exits the marsupium is, in all essential respects, a functional isopod. It requires no further transformation. Its development was completed before it ever entered the world. Manchae emerge from the brood pouch with 6 of their eventual 7 pairs of legs already present and functional. The 7th pair, located at the posterior end of the perion, does not develop until after the first molt, the first shedding of the exoskeleton that occurs in early juvenile life. The mancha is therefore functional immediately upon emergence, capable of walking and navigating independently, despite the temporary absence of the final leg pair. The missing pair develops in the normal course of early growth and is present by the time of the second molt. The sequence reflects the completeness of in-pouch development, while also indicating that the final anatomical feature of the adult form is acquired through post-emergence maturation rather than within the marsupium. The gestation period for giant isopod eggs, the duration from fertilization to emergence of the manchae from the marsupium, has not been precisely established. Available evidence from captive specimens and field-collected data suggests the period is extended, likely measured in months and possibly exceeding 1 year in some cases. The difficulty in establishing precise figures reflects the fundamental challenge of studying deep-sea reproductive biology. Animals collected from depth for study are often stressed, their developmental timing disrupted, and controlled conditions in aquaria do not fully replicate the thermal and pressure environment of the deep sea floor. What is understood is that isopod reproduction, like most other aspects of the animal's biology, operates on a slow and extended timescale. Current understanding of giant isopod reproductive biology indicates that females likely cease feeding entirely during the brooding period. All available energy reserves are directed toward maintaining the marsupium and supporting the development of the eggs it contains, rather than toward the female's own metabolic maintenance beyond the minimum required to sustain life. This reproductive investment is substantial, particularly given that the isopod's energy reserves are already limited by the infrequency of food availability in the deep sea. A brooding female is simultaneously managing her own survival and the full developmental cost of her clutch without any food intake for a period that may extend over many months. The lifespan of the giant isopod in the wild has not been directly measured, but estimates derived from growth rate data in captive specimens suggest individuals may live between 8 and 15 years. This is a relatively long lifespan for a marine invertebrate of comparable size. Extended lifespan is consistent with the isopods' slow metabolism, cold habitat, and conservative growth pattern, factors that collectively reduce the rate of cellular wear and the accumulation of physiological damage over time. The isopod grows slowly, reproduces infrequently, and lives for a substantial period relative to its body size. These traits form a coherent life history strategy suited to the energetic and ecological constraints of the deep sea. Male and female giant isopods are believed to encounter each other infrequently, with mating events likely facilitated by chemical signals, pheromones released into the water that indicate reproductive readiness and allow individuals to locate one another across the seafloor. In an environment where individuals typically move alone across large areas of substrate with low population densities, chemical signaling provides the primary mechanism for bringing potential mates into contact. The specifics of isopod mating behavior have not been extensively documented, partly because the behavior is difficult to observe at depth and has been rarely replicated under controlled conditions. What is understood is that contact between individuals for reproductive purposes is infrequent and mediated primarily through chemosensory detection. Giant isopods reproduce slowly and infrequently, with small clutch sizes, extended brooding periods, and long intervals between reproductive events. This life history pattern contrasts with the high-volume, low-investment reproductive strategies of most shallow-water marine invertebrates, which release large numbers of small eggs and rely on probability for offspring survival. The isopod's approach reflects the ecological conditions of the deep sea, where food availability is uncertain, population density is low, and survival to reproductive age depends on the ability to sustain life through extended periods without resources. In that context, investing heavily in a small number of well-protected offspring is a more reliable strategy than producing many and releasing them to an environment that offers no guarantee of their survival. Giant isopods have evolved a metabolic system capable of downregulating substantially during periods of food scarcity. When food is unavailable for extended periods, the isopod's cellular processes continue at a reduced rate, sufficient to maintain basic physiological function but demanding far less energy than normal active metabolism. This is not a state of dormancy or sleep. The animal remains responsive to its environment throughout. What changes is the rate at which it burns its stored reserves. The isopod's documented fasting capacity, surviving without food for years in captive settings, is a direct expression of this metabolic flexibility. The system was shaped by millions of years of feast or famine conditions on the deep sea floor. The exoskeleton of the giant isopod is composed of two primary structural materials: calcium carbonate, which provides hardness and rigidity, and chitin, a natural polymer that contributes flexibility and prevents the shell from becoming brittle under the mechanical stresses of deep sea pressure. Chitin is one of the most abundant organic compounds on Earth, found in the shells of insects, the cell walls of fungi, and the exoskeletons of many other arthropods. In the isopod's shell, calcium carbonate and chitin are present in proportions that produce a material combining structural stiffness with enough elasticity to absorb and distribute cyclic stress without fracturing. The combination has proven effective under pressures that would destroy most other biological structures. The near-freezing water temperature of the deep sea acts as a metabolic regulator for the isopod, slowing every biochemical process, reducing the rate at which cells consume oxygen and produce metabolic waste, and lowering the overall energy demand of maintaining a living body. For warm-blooded animals, cold is a physiological threat that requires active management. For the giant isopod, cold is the operating condition around which its entire biology has been calibrated. The animal's enzyme systems, cellular membranes, and metabolic pathways all function at reduced temperature. Cold does not slow the isopod in the way it would slow a mammal placed in the same environment. It is the thermal context in which the isopod functions normally. The large compound eyes of giant isopods are adapted for detection in near-total darkness. They are calibrated not to resolve fine visual detail, a function that requires substantial ambient light, but to register any trace of light present in their environment, including the bioluminescent emissions of other deep-sea organisms. Researchers believe the eyes may be capable of detecting the faint light produced by anglerfish lures, alarmed crustaceans, or bacterial bioluminescence on organic material nearby. In the deep sea context, any light signal carries potential information about food, predators, or conspecifics. The isopod's visual system appears to be organized around detecting those signals rather than constructing a detailed image of the surrounding environment. Isopods lack a swim bladder, the gas-filled organ that allows bony fish to regulate buoyancy and maintain depth without active swimming effort. The absence of any gas-filled internal cavity means that the isopod is not subject to the pressure-related buoyancy changes that affect fish when moved rapidly between depths. Swim bladders compress at depth and expand toward the surface, creating serious physiological risk for fish moved quickly through a large depth range. The isopod, containing no compressible gas spaces, is immune to this mechanism. It is simply dense, and its density makes it compatible with the full range of pressures present across its habitat depth range without any physiological adjustment required. The segmented body plan of the isopod gives it a degree of maneuverability over irregular terrain that would be unavailable to an animal with a rigid, unsegmented exterior. Each body segment can flex at its joint with the adjacent segment, allowing the body as a whole to conform to the shape of the surface beneath it as the animal moves. This independent articulation means that the isopod can maintain stable contact with the seafloor across changes in substrate geometry, navigating over ridges, into depressions, and across transitions between sediment types without losing traction or stability. The flexibility of the segmented body is one of the core functional advantages of the arthropod body plan, and the isopod expresses it with particular effectiveness in the complex topography of the benthic zone. When an isopod curls into a defensive sphere, the overlapping edges of its body plates come together along their margins to form a near-continuous seal around the animal. The soft internal structures, gills, mouthparts, legs, and in brooding females the marsupium and its contents, are enclosed within the shell with minimal exposed surface area. A predator attempting to penetrate the sphere must apply force to a curved, segmented surface that distributes that force across multiple plate edges simultaneously. The structural resistance of the curled isopod is therefore considerably greater than the resistance of any individual plate in isolation. The sphere is not just a shape, it is an integrated pressure distribution system activated as a defensive posture. The chemoreceptors of giant isopods, distributed across the antennae and over the surface of the body, are believed to be exceptionally sensitive instruments, capable of detecting dissolved organic compounds at concentrations that would be difficult to measure with standard analytical equipment. In the deep sea, where a food source may be located hundreds of meters away across open sediment, the ability to detect and track a chemical gradient through the water is the primary means by which the isopod locates nutrition. The sensitivity of these receptors reflects millions of years of selection pressure in an environment where the ability to detect trace concentrations of organic material from a distance is directly tied to survival. Some isopod species within the broader order have been documented in proximity to hydrothermal vents, seafloor features that release superheated chemically complex water from within the oceanic crust. Vent environments present conditions that are extreme even by deep sea standards. Elevated temperatures, high concentrations of sulfur compounds and heavy metals, and pH levels outside the range found in the surrounding ocean. The presence of isopods in or near these environments indicates a tolerance for chemical and thermal variability that extends beyond simple adaptation to cold, stable conditions. The broader isopod order has demonstrated, across its many species, a capacity to inhabit the full range of conditions that the deep ocean floor can produce. The pale coloration of giant isopods is, at least in part, a metabolic consequence rather than an active adaptation. Producing and maintaining dark pigmentation requires biological resources. The synthesis of pigment molecules consumes energy and raw materials that the isopod's deep-sea environment provides in limited quantities. In the absence of any functional role for coloration in a lightless habitat, the cost of pigment production is an unnecessary expenditure. Natural selection in the deep sea has therefore not maintained dark pigmentation in Bathynomus giganteus. The pale grey and lilac tones of the animal reflect the absence of pigment rather than its active production. a body conserving resources by not manufacturing what its environment makes irrelevant. Giant isopods are a recognized example of deep-sea gigantism, the tendency for animals in the deep ocean to grow substantially larger than closely related species in shallower water or on land. The common pillbug, the terrestrial relative of the giant isopod, rarely exceeds 2 centimeters. Bathynomus giganteus regularly reaches 30 to 40 centimeters. Multiple factors are thought to contribute. Cold temperatures, slow metabolism, and extend the growth period over a longer lifespan. Reduced predation pressure in the deep sea allows individuals to survive long enough to accumulate size. High dissolved oxygen levels in cold water can support larger metabolic demands, and larger body size provides greater energy storage capacity in a feast or famine food environment. No single factor fully accounts for the size difference, but the combination produces a consistent and well-documented biological pattern. Deep-sea gigantism is a well-documented biological pattern in which species living in deep ocean environments consistently attain larger body sizes than closely related species in shallow water or terrestrial habitats. The pattern has been recorded across numerous and taxonomically unrelated animal groups: giant squid, giant tube worms, giant amphipods, giant sea spiders, and giant isopods among them. The consistency of the pattern across such a broad range of organisms suggests that the deep ocean environment, or the combination of factors that define it, exerts a selective pressure favoring larger body size over evolutionary time. The mechanisms involved remain an area of active scientific inquiry, with multiple contributing factors identified and no single explanation sufficient on its own. Bathynomus giganteus is a defining example of deep-sea gigantism, reaching sizes approximately 20 times greater than its closest terrestrial relative, Armadillidium vulgar, the common pillbug. Both animals share the same fundamental body plan, the same articulated segments, the same capacity for conglobation, the same arrangement of limbs and sensory organs. The difference in scale between them is a record of what millions of years of deep-sea life does to a shared body plan, not altering its essential structure, but expanding it. The relationship between the two species, visually incongruous as it is, reflects the degree to which environment shapes physical form over sufficiently long periods of time. Several hypotheses have been advanced to explain deep-sea gigantism, and the current scientific consensus is that the phenomenon is likely driven by a combination of contributing factors. Cold temperatures slow metabolic rate and extend the growth period, allowing individuals to accumulate size over longer lifespans than would be possible in warmer, faster metabolizing populations. Reduced predation pressure in the deep sea allows animals to survive long enough to reach exceptional sizes. High dissolved oxygen concentrations in cold, deep water may support the metabolic demands of larger bodies, and the feast or famine character of deep sea food availability may favor larger body sizes capable of storing more energy between feeding events. These factors interact and reinforce one another in ways that researchers continue to examine. Among the more significant recent additions to isopod taxonomy is Bathynomus raxasa, formally described in 2020 from specimens collected in the eastern Indian Ocean. The species name raxasa means giant in Indonesian, a fitting designation for an animal that rivals Bathynomus giganteus in its physical dimensions and represents one of the largest isopod species currently known to science. The description of B. raxasa in 2020 added a new major species to the genus Bathynomus and extended the documented geographic range of supergiant isopods into the Indian Ocean. Bathynomus giganteus retains its status as the historically dominant species in terms of total bulk and the depth of available scientific documentation. The ongoing discovery of new giant isopod species reinforces a broader point about the state of deep-sea taxonomy. Large, physically substantial animals remain undescribed in ocean regions that have been studied for decades. Bathynomus yucatanensis, formerly described in 2022 from specimens collected in the Gulf of Mexico, is one recent example. The Gulf of Mexico is among the most historically studied deep water bodies in the Western Hemisphere, and yet a distinct species of giant isopod within it went unrecognized until the 21st century. The implication for less examined ocean regions is significant. The inventory of large benthic crustaceans is not complete. New species continue to be identified as survey methods improve and sampling coverage expands. Some researchers propose that deep-sea gigantism in isopods is reinforced, in part by the ecological dynamics of food availability in the benthic zone. In an environment where substantial food arrives infrequently and unpredictably, larger body size provides a competitive advantage at feeding events. A larger animal can consume more at a single feeding and retain more stored energy to sustain itself through the intervals between events. Under this interpretation, the giant isopod's size is not solely an incidental consequence of cold temperature and longevity, but partly a functional adaptation to a specific ecological pattern. Large, rare food inputs requiring large, efficient storage capacity in the animals that exploit them. Despite their size relative to terrestrial isopods, giant isopods move without aggression and without any behavioral characteristic associated with the dominance their scale might otherwise suggest. Their movement is slow and deliberate, their interactions with other animals non-confrontational, and their response to unfamiliar stimuli measured rather than reactive. The size of Bathynomus giganteus accumulated gradually over an immense span of time through the slow action of metabolic and ecological selection pressures in the deep sea. It was not acquired through competition or territorial behavior. It is simply what the animal has become after millions of years of living where it lives, at the pace it lives, slowly, continuously, and without apparent urgency. Isopods are bilaterally symmetrical. The left and right sides of their body are mirror images of each other, leg for leg, plate for plate, antenna for antenna. This symmetry is functional rather than incidental. It distributes weight and movement evenly across the body, making locomotion as efficient as possible. Bilateral symmetry is a defining feature of most animal groups and reflects a deep evolutionary logic about how bodies move through space. In the isopod, that logic is expressed with particular clarity. The body is organized around a central axis, balanced on either side, adapted for steady, forward movement across the ocean floor. Deep-sea isopods breathe through structures called pleopods, paddle-shaped appendages on the abdomen that function as gills in aquatic species. In the cold, pressurized depths of the ocean, oxygen is present but extracted slowly, and the pleopods are well adapted to that constraint. They move in a gentle, continuous rhythm beneath the body, drawing oxygenated water across the gill surfaces as the animal rests or moves. Breathing, for the isopod, is a constant and low-energy process, a quiet, ongoing exchange with the water that surrounds it, running beneath all other activity without interruption. Deep-sea isopods are invertebrates. They have no internal skeleton and no backbone. Their structure comes entirely from the outside, from the curved, overlapping plates of their exoskeleton, which simultaneously protects, supports, and defines the animal. Where vertebrates carry their structural framework internally beneath soft tissue, the isopod wears its architecture externally. The exoskeleton is not a secondary feature or a protective layer added over some other structure. It is the structure. The shell is what holds the isopod together, gives it its shape, and allows it to function in one of the most mechanically demanding environments on the planet. The giant isopod, Bathynomus giganteus, reaches lengths of 30 to 40 centimeters in most documented specimens, roughly the size of a large shoe. A small number of individuals have been recorded approaching 50 centimeters, approximately the length of a small house cat. Those specimens of that size are exceptionally rare and represent the outermost edge of what the species achieves. By any measure, this makes Bathynomus giganteus one of the largest crustaceans living on the deep ocean floor, a scale that remains difficult to fully appreciate until a specimen is placed alongside a familiar object for comparison. The body of an isopod is composed of a series of overlapping, articulated segments. Each segment is connected to its neighbors by flexible joints, allowing the body to curve, flex, and even curl completely inward while maintaining structural integrity. The segments are stacked and shaped in a way that distributes mechanical force across multiple surfaces simultaneously. This design has remained functionally unchanged for hundreds of millions of years, a testament to its reliability across an extraordinary range of environments and conditions. The articulated body is neither rigid like stone nor soft like flesh. It occupies a structural middle ground that has proven highly durable over evolutionary time. Isopods have a rigid dorsal shield called a cephalon that protects the head and all primary sensory organs, the eyes, the antennae, and the mouthparts. The cephalon is thick and curved, shaped to deflect impact and reduce the exposure of the most functionally important structures in the animal's body. Behind it, the eyes gather traces of light in near-total darkness. The antennae sample the chemical composition of the surrounding water, and the mouthparts remain protected until needed. The cephalon is the forward-facing shield of the isopods architecture, the first surface to meet the environment and the one most heavily reinforced. Isopods have 7 pairs of legs, 14 in total, each pair attached to a separate body segment called a perionite. The legs move in a coordinated sequence that produces a slow, steady rolling motion across the seafloor. Each leg lifts and places in turn, producing a ripple of movement that passes from front to back along the length of the body. This gait is energy efficient and well suited to the low metabolic demands of deep sea life. 14 legs operating together allow the isopod to maintain stable contact with the seafloor across irregular surfaces and under the variable pressures of deep water currents. The giant isopod entered the formal scientific record in 1879 when specimens were recovered during deep-sea dredging operations in the Gulf of Mexico. The nets used in these expeditions were dragged along the ocean floor and hauled back to the surface with whatever organisms they collected. Among those organisms in that year was a large, heavily armored crustacean that matched nothing in the existing scientific literature. Alphonse Milne Edwards, the French zoologist who examined and described the specimens, placed it formally in the record under the name Bathynomus giganteus. The description he published established the first documented baseline for the species, its physical form, its size, and its general characteristics as a deep-sea animal. The discovery of the giant isopod in 1879 had implications that extended beyond the species itself. At the time, a widely held view in the natural sciences held that the deep sea was largely devoid of animal life, too cold, too dark, and under too much pressure to support biological communities of any substance. The giant isopod was part of a body of evidence accumulating through the second half of the 19th century that directly contradicted this position. Large, structurally complex animals were being recovered from depths previously thought to be uninhabitable. The deep sea was not empty. It supported life that was unfamiliar in form but clearly adapted to conditions that, until then, had been assumed to preclude it. Naturalists who encountered giant isopod specimens in the collections of 19th century deep-sea expeditions responded with a mixture of scientific interest and evident surprise. The specimens were described in contemporary accounts as sea bugs of disproportionate scale, creatures bearing the form of the common pill bug, but at a size that appeared to belong to a different order of nature entirely. The natural science of the era did not have well-established frameworks for large deep-sea invertebrates of this type. New descriptive categories had to be developed, and existing assumptions about the relationship between depth and biological diversity had to be revised. The giant isopod was among the first deep-sea animals to force that revision. Today, giant isopods are studied using a range of technologies suited to the operational challenges of deep-sea research. Crewed submersibles carry researchers to depth for direct visual observation. Remotely operated vehicles provide video documentation and allow physical collection of specimens without requiring human presence at depth. Baited camera traps and collection nets deployed from surface vessels on long cables provide population-level data and specimens for laboratory analysis. Each method produces a different category of information: behavioral, morphological, genetic, or physiological, and the combination has substantially expanded scientific understanding of the species over the past several decades. Research continues, and significant gaps in documented knowledge remain. Video footage of giant isopods feeding at whale carcasses on the deep sea floor, captured by remotely operated vehicles during deep sea biological surveys, became some of the most widely viewed recordings from the deep ocean during the early period of internet video distribution. The footage showed dense aggregations of pale, armored animals moving with deliberate efficiency across the surface of large carcasses in total darkness at depth. For a broad public audience, the footage provided the first direct visual evidence of the ecological activity of the benthic zone. The images were striking and unfamiliar enough to generate sustained public interest in deep-sea biology at a time when such environments had received relatively little mainstream attention. The biological characteristics of giant isopods have attracted scientific interest beyond the field of marine biology. The isopods' documented capacity to survive for years without food has relevance to research into metabolic diseases and the physiological mechanisms of starvation tolerance in animals. The structural properties of the chitin-reinforced exoskeleton have potential applications in materials science research focused on lightweight, pressure-resistant composites. The animal's resistance to extreme hydrostatic pressure is of interest to researchers studying biological function under high-pressure conditions more broadly. The giant isopod is, in this sense, not only a subject of ecological study but a biological reference point for a range of questions in applied and basic science. In Japan, the giant isopod has developed a notable public profile that is unusual for a deep-sea benthic crustacean. The animals are displayed in public aquariums, where they consistently draw significant visitor attention. They appear as subjects of commercial merchandise, plush toys, phone accessories, printed materials. They generate periodic trending activity on Japanese social media platforms, and a dedicated enthusiast community follows news of new species descriptions and newly arrived captive specimens with sustained interest. The reasons for the isopod's particular resonance with the Japanese public have not been formally studied, but the combination of unusual appearance, genuine biological strangeness, and accessible aquarium display appears to have produced a durable popular interest in the species. Among the most extensively documented animals in the history of public aquarium keeping is a giant isopod held at Toba Aquarium in Japan, catalogued by staff as number 1. The animal arrived at the facility in 2007 and refused all food offered from the time of its arrival. The fast continued for 5 years and 4 months before the animal died in 2014. Aquarium staff monitored its condition throughout, offering food at regular intervals without success. The case received international scientific and media coverage, and number 1 became the most publicly recognized individual isopod on record. Its fast remains the longest documented fasting period for a giant isopod in a controlled environment, and the reasons for it were never fully explained. New species of deep-sea isopod continue to be formally described at a regular rate, produced by expanding deep-sea survey programs, advances in collection and imaging technology, and increased scientific attention to benthic invertebrate taxonomy. Each new species description adds to the documented range and diversity of the Bathynomus genus and the broader isopod order. The frequency of new descriptions in recent decades indicates that the existing species inventory is incomplete and that the actual diversity of the group remains greater than what has been formally documented. The deep ocean continues to yield undescribed organisms, including large, morphologically distinctive species, as survey coverage and analytical methods improve. Isopods belong to the class Malacostraca, the largest class of crustaceans, encompassing more than 25,000 known species. Malacostraca includes crabs, lobsters, shrimp, krill, mantis shrimp, and amphipods, among many others. Members of this class occupy habitats ranging from the deepest ocean trenches to freshwater rivers, coastal margins, and terrestrial environments. The class represents one of the most ecologically diverse and geographically widespread groups in the animal kingdom. Within it, the isopod order is among the most species-rich, having colonized a broader range of habitats than almost any other crustacean group. The giant isopod is the deep-sea expression of this broader lineage, the form it takes in the most extreme subset of its range. Despite the apparent strangeness of their appearance to human observers, isopods share evolutionary ancestry with many familiar animals: the shrimp on a dinner plate, the crayfish in a freshwater stream, the barnacle attached to a dock piling. All are crustaceans, and all share a common ancestor with the isopod at some point deep in evolutionary history. The body plans have diverged substantially over hundreds of millions of years of separate evolution, but the underlying biological heritage is shared. The giant isopod is not an alien organism in any meaningful biological sense. It is a distant relative of well-known animals, shaped by the particular demands of a particular environment into a form that reflects those demands, rather than a fundamental departure from the crustacean lineage. The evolutionary lineage of isopods extends back to the Cambrian seas more than 500 million years ago, when the arthropod body plan, segmented body, external skeleton, jointed appendages first appeared in the fossil record. The isopod's current form is not identical to those ancient ancestors, but it is recognizably related, a modern elaboration of a structural template that has remained fundamentally viable for 500 million years. The persistence of the arthropod body plan across this time span is among the most compelling evidence in biology for the durability of certain structural solutions. The isopod does not represent a departure from that ancient design. It represents one of its current expressions, adapted to a specific set of conditions by the particular history of its lineage. Giant isopods perform a critical ecological function in the benthic zone as decomposers and nutrient recyclers. By consuming organic material that sinks from the upper ocean and processing it through their digestive systems, they return carbon, nitrogen, and other nutrients to the sediment in forms accessible to the smaller organisms that occupy the base of the deep-sea food web. Without this processing activity, organic matter would accumulate on the seafloor in forms unavailable to most of the organisms that depend on it, altering the chemistry and productivity of the benthic environment. The isopod's ecological role is not visible or dramatic. It is the quiet, continuous work of an animal doing what its biology has always done, in a place that has always needed it done. The existence of giant isopods, and the broader community of organisms they belong to, is a significant counter to assumptions about what life requires. Common intuitions associate biological vitality with warmth, light, and nutritional abundance. The giant isopod lives in cold, darkness, and scarcity, and has done so continuously for hundreds of millions of years. Its existence is not a marginal exception to a rule that life requires favorable conditions. It is evidence that the parameters within which life is possible are considerably broader than surface world conditions suggest. The deep sea, at its most inhospitable, supports animals, adapted, functional, and persistent animals, that have never required any of the conditions we tend to associate with life's vitality. Giant isopods are frequently cited by researchers as examples of deep-sea resilience, not in the sense of dramatic survival against odds, but in the sense of sustained, low-energy persistence over an immense span of time. The isopod does not resist the conditions of the deep sea. Its metabolism, body structure, reproductive strategy, and behavioral profile are all organized around the specific parameters of that environment. Resilience, in the isopod's case, is not the capacity to endure adversity. It is the result of having become, over millions of years, precisely what the environment called for. An animal calibrated to the deep sea's constraints so thoroughly that those constraints no longer function as constraints at all. The scientific study of giant isopods has continued to produce findings relevant to fundamental questions in biology about metabolic flexibility, structural adaptation to extreme pressure, reproductive investment strategies under resource limitation, and the mechanisms by which body size scales with environmental conditions over evolutionary time. Each study of the isopod adds information not only about the animal itself, but about the biological principles it demonstrates. The giant isopod is, in this sense, a research subject with implications that extend well beyond its own species, a reference point for understanding how life responds and adapts to the full range of conditions that the planet's environments can produce. At this moment, in the Atlantic Ocean, in the Gulf of Mexico, and in the eastern Indian Ocean, giant isopods are moving across the deep sea floor. Their antennae trail through water that is a few degrees above freezing, sampling the chemical composition of the current for any trace of organic material nearby. Their 14 legs place with measured regularity against cold sediment. No light reaches them. No sound as we understand it accompanies their movement. They have no awareness of being studied, no awareness of their own evolutionary history, no awareness of the scientific literature that has accumulated around them over the past century and a half. They are present in the dark, at depth, doing what this lineage has done for 300 million years, moving forward slowly across the floor of the world. And slowly, we rise, back through the dark water, back through the cold and the pressure, back to the surface where you are lying still and breathing quietly. The isopod remains below, patient, unhurried, ancient, alive. It has no need of light, no need of warmth, no need of being known. It needs only the dark and the cold and the slow floor of the world beneath it. Let that stillness carry into your own body now. Somewhere below everything, something is at rest. Something has always been at rest. You can be too.